Hello, how's it going? Today I'm going to present on E2F1's predicted structure and the role of some anchoring amino acids within E2F1 in its interaction with cyclin F. My name is Joshua Harvey, and let's go ahead and get started. So first, I just want to talk about E2F1 and cyclin F within the bigger picture of the cell. So cyclin F fits within the SCF complex. And so the SCF complex is a complex of proteins contained largely of the three main proteins, an F-box protein, a skip1 protein, and a col1 protein. And the purpose of the SCF complex is to ubiquitinate its substrates. And so ubiquitination is just the addition of ubiquitin groups to a protein, which marks it for degradation um, and disables the function of the protein. And so, if you've heard of cyclins before, you might be thinking that is not really what I think a cyclin usually does. And so it's interesting to look at the difference between cyclin F and some other normal other cyclins that we might know about, such as cyclin A. And so if you look over to the images on the right side of the screen, you can see that some normal cyclins, such as cyclin A, are known to phosphorylate their substrates instead of add ubiquitin groups to them. And so this phosphorylation, in contrast to the ubiquitination that has been shown in the SCF complex with cyclin F, um, it might activate these different substrates by adding phosphate groups to them instead of degrading them by adding ubiquitin groups to them. And so let's look a little bit more about what E2F1 is outside of just a substrate for cyclin F. And so E2F1 acts as an activator that promotes movement through the cell cycle through the G1S transition. And it is also situated within a larger family of E2F1 proteins that act as transcriptional regulators within the cell. So now I just want to take the time to look a little bit more deeper into the specifics of the interaction between E2F1 and cyclin F. So E2F1 is known to have what is called a psi motif, and this specific psi motif that is known to interact with cyclin F comes um, between amino acids 85 and 97 in the primary sequence of the protein. And so a psi motif is just three amino acids. The first one is in arginine. The second one can be any different amino acid, and the third one is a leucine. And so this specific psi motif, which is the second psi motif within E2F1, so I'm going to call it psi motif 2 throughout the rest of this presentation, is known to interact specifically with the amoral site on cyclin F. And so the amoral site is contained of the amino acids M, R, Y, I, and L, and it's very similar to the M-rail sequence in cyclin A. Um, and the Emerald site for cyclin F is known to be located between amino acids 304 and 318. And so the picture on the left is just a mapped interaction between the psi motif 2 of E2F1 and the Emerald site of cyclin F. And so since it's known that the psi motif 2 of E2F1 interacts with the M rail of cyclin F, it starts to beg some different questions behind how this interaction works. And so, since there are multiple different psi motifs within E2F1, I started to wonder why specifically does the Emerald site locate to psi motif 2 within E2F1 and not psi motif 1 or psi motif 3? Um, Then, like more broadly, I also was kind of wondering how does cyclin F know specifically how to interact with E2F1 and not a psi motif of another protein? Because there are so many different proteins, and psi motifs can be very common throughout different proteins. Um, just because it's a small amount of amino acids, and there's going to be an arginine followed by a random amino acid followed by a leucine throughout other proteins. And so why specifically does it interact with E2F1 and why specifically does it interact with this specific Cy motif in E2F1. And then the M rail site of cyclone F is very similar to the M rail site in cyclone A. The only difference is that this Y, um, shown here in the middle of the emerald side of cyclone F, is an A in cyclone A. And so how does the emerald site know, or how does E2F1 know to interact with the emerald site of cyclone F and not the emerald site of cyclone A? And so see, these are some of the questions that I've been thinking about in trying to answer through this research. And then so continuing, trying to solve this structure and solve some of these questions. Um, the mapping of interactions between proteins in lab, in vitro, can be very laborious. It can be very long. It can be very difficult. And so the methods that I've chosen to go with to investigate some of these questions, I have chosen to use AlphaFold 3, which is a predictive AI powered analysis tool. You kind of plug in different amino acid primary sequences and it will spit out an IPTM score, which is a confidence score, and a predicted structure for this protein interaction. Um, and so this is quicker and more convenient than testing in the lab, and it allows for more testing in less time. But there is a downside. These interactions are only predictive interactions, and so we can't tell for certain whether this will actually be happening in the cell. And so the first major question that I was kind of wondering when I started to tackle these questions was, can AlphaFold accurately predict this interaction? And so it's known that the Cy motif 2 localizes to the embryo in the literature. And so I was thinking that if Cy motif 2 localizes to the embryo in the predicted um, AlphaFold structure, and an IPTM score was very high, the confidence score is very high, then AlphaFold is likely to be able to predict this interaction very well. And so as you can see in this structure on the slide, the Psi motif in blue is shown directly interacting with the MREL site in orange. And so it was concluded that AlphaFold can accurately predict this interaction between E2F1 and the MREL. So next I'm kind of wondering, we know that AlphaFold can accurately predict the Psi motif to localize to the MREL in E2F1 and cyclone F. But I was wondering, are there any predicted closely interacting amino acids around those sites that might help facilitate this interaction? Um, and so from the original interaction from AlphaFold between E2F1 and cyclone F, I used PyMol to label different amino acids within 5 angstroms of each other. So that's an amino acid on E2F1, an amino acid on cyclone F, and are they closely interacting within 5 angstroms of each other? And so when labeling these, we can see on the picture on the slides, E2F1 is shown in yellow. The parts of E2F1 that interact with cyclone F are shown in purple, closely interact, and the parts of E2F1 that closely interact with the emerald site are shown in a lighter purple, more lavender color. And then also cyclone F as a whole is shown in light blue, and then the parts of cyclone F that interact specifically with E2F1 are shown in red, and the parts of cyclone F that interact with Psi motif 2 are shown in a more lighter brown color. And so from this, it was found that amino acids outside of the Psi motif 2 and the M-rail closely interact with the different proteins and with the Psi motif and with the M-rail site. And so this leads me to do some more mutations around these closely interacting amino acids, specifically in E2F1, to investigate how they might affect binding. And so the main question I'm trying to answer with this trial is, do the specific amino acids that are predicted to closely interact affect binding, and do some of them not? Are some important while others aren't? And so to investigate this, there were many mutants made, 18 in this trial, in which um, some of these closely interacting amino acids, sometimes the cy motif were mutated from the amino acid they were into an alanine, which is very small and non-interacting. And then they were predicted in AlphaFold. And so the graph on the left, you can see the y-axis shows IPTM scores, or confidence score, and then the x-axis shows which mutant is being tested. And so an IPTM score, as previously stated, measures whether the structure is likely to be true or not. It's generally accepted that an IPTM score above or at zero point 0.5 is a true structure. Um, and this can be kind of interpreted in this data that the psi motif and the MREL are still interacting if there's a strong predicted interaction. And then once the IPTM scores falls below zero point 0.5, AlphaFold is no longer to predict this interaction. And so this can kind of be seen as the interaction between the psi motif 2 and the MREL has been broken. And so from this data, it was found that as little as mutating as little as six closely interacting amino acids on either side of psi motif 2, which is a total of 12, breaks the interaction between the psi motif and the MREL, while going down a little bit further to 5 amino acids on the other side, or a total of 10, still allows the interaction to occur. This can be seen in mutant 12, in which 6 amino acids are mutated, and the IPTM score is below zero point 0.5, and mutant 11, in which 5 closely interacting amino acids on either side of the psi motif 2 are mutated, and the interaction is still predicted to occur. This shows specifically, in the larger context, that there are amino acids that help contribute to the facilitation of the interaction outside of the psi motif in E2F1. And that some of the that these are just a few of the predicted closely interacting amino acids among the larger group of predicted amino acids, that were predicted by AlphaFold originally. And it is important to note too that in mutant 5, when just the psi motif 2 is mutated, the interaction is not likely to occur. And this is a good control for this. Now, going further from just the 6 amino acids found to be important to binding on either side of the psi motif, um, I was hoping to extrapolate and get down more to have some less amino acids to really see which ones are important and which ones aren't. And so the main question I'm trying to answer here is, do all 12 amino acids found break binding when knocked out, or are they all contributing to binding? And so from mutant mutant 12 in um the previous slide, I made mutants one by one of mutating one amino acid for each mutant. And so this goes one by one down, starting at the look closest to the N terminus and finishing closest to the C terminus. Um, and so again, it was regarded, if the IPTM score is above 0.5, then okay, I'm gonna backtrack about 30 seconds because I explained that incorrectly. Um, and so for this trial, amino acids are added back from the mutant 13 in, sorry, from mutant 12 in the previous experiment, in which six amino acids were mutated on either side of the Si motif. Six closely interacting amino acids were mutated on either side of the Si motif. And so these already mutated amino acids were returned one by one, and the trial was run again. And so mutant one corresponds to returning the amino acid for like closest to the N terminus, and then mutant 12 corresponds to returning the amino acid closer to the C terminus. And so, kind of quantifying these results, if the IPTM score is above 0.5, then binding is considered to be restored, and this amino acid is considered to be important to facilitate binding. Now, if the IPTM score remains below 0.5, as like in mutant 12 from the last slide, the interaction remains to be unable to be predicted in AlphaFold, and the binding is still considered to remain broken. And so from this, mutants 10 and 11 um, predict that the amino acids aspartic acid and glutamine, as shown in the primary sequence on the slide, are not important to facilitating binding. And then mutant 13, in which both of these are mutated together, confirms that they are both not important when returned together. Now, continuing on this thought, I ran another trial in which um, mutant 13 from the previous slide was used. And again, mutants were made in which one true amino acid was returned to the mutated amino acids, starting at the N terminus and going all the way to the C terminus. And so from this, it was found in mutants 1, 9, and 10 that glycine, glutamic acid, and tyrosine were not deemed to be important. And this can be seen again in the primary sequences at the bottom. And so mutant 11 specifically then combines all of these amino acids and returns all of them to see if they're all important together. Um, and mutant 11 predicts that they might all be important together since the IPTM is above 0.5. Mutant 12 also then predicts that G and E together might be important if they're both missing. And then mutant 13 shows us that if the glutamic acid and the tyrosine are both mutated together, they're not important for binding. And mutant 14 predicts that if the glycine and the tyrosine are both mutated together, then they could be important for binding. And so this leaves us with two possible constructs when trying to figure out the least amount of amino acids that breaks binding. And so these constructs are shown as their primary sequences below the graph. Um, and so one mutates only the glycine from the previous mutant 13 of the previous slide, and this is mutant 1, and the other mutates both the glutamic acid and the tyrosine, and this is mutant 13. And so then going on, just trying to confirm that there are no more amino acids that um, are not important for binding. IPTM scores were obtained, again, going one by one, returning these amino acids from mutants 1 and 13 from the previous slide. And it's found that all remaining amino acids were predicted to help facilitate binding between the Si motif 2 and the M rail because their IPTM scores remained high. Or were returned to being high, and so predicted the interactions occurred together when they were returned. And so just to reiterate, because the interaction between Simotif 2 and the MREL in E2F1 and cyclone F is not completely understood. It was I hypothesized that there might be some amino acids surrounding these sites that could contribute to the facilitation of this interaction. And so the first finding at the very beginning is that I found that AlphaFold is very reliable in predicting the interaction between E2F1 and cyclin F, and more specifically between Cy-motif 2 and the emeryl site. And this predicts a number of closely interacting amino acids between the proteins. And then furthermore, as we can see between these two primary sequences up on the screen, when knocked out a group of 9 amino acids, these alanines in blue, highlighted in blue, and a group of 8 amino acids, and then this is below, also highlighted in blue, still contain low IPTM scores and are found to break the binding and the interaction between Cy-motif 2 and the emeryl in the presence of both of these Cy-motif and emeryl still being intact. And so from there, we can jump to testing these more in lab in vitro, and these trials in AlphaFold have made it so that we can not waste materials and not waste time trying to figure out how specifically these interact, because there are a lot of different mutants that were predicted and tell us a lot about the interaction between these proteins, and no science actually had to be done in the lab. And now having a better understanding of what might be happening between these two proteins and in this interaction, this can then be tested in the lab setting. So just want to thank you guys for bearing with me through this presentation, and I hope that you guys learned a little bit and found it interesting, and have a great day.